Hey everyone. So up until now, we have treated biological data like sentences in a book. But in the cell, proteins are not flat. They fold into intricate in these beautiful 3D shapes that determine exactly how they function. So today, we are stepping into the world of structural bioinformatics. We are going to learn how to read PDB files, which contain the 3D coordinates of every single atom in a protein, and we will use BioPython to navigate these structures and even perform some 3D geometry. So let's get started. So what is a PDB file basically? It's a standard format for 3D structures. It's essentially a giant list of atoms, and then each file tells you the atom's name, the amino acid it belongs to, and its XYZ coordinates in a 3D space. So BioPython organizes this data into a hierarchy called SMCRA, structure, model, chain, residue, and atom. So think of it like a folder system. A structure contains models, which contains chains, and then which contains residues, which are also amino acids, and then which finally contains atoms. So first we will need to download a structure. You can go to this website, rc uh, sb.org, and you can search for any protein there. For this examples, we're going to use one fat. This is a plant protein. So let's load it using the PDB parser. Right. So yeah, let's get started. From the library that we will import is bio.pdb import pdb parser. So we will continue. Parser is equal to pdb parser. This will initialize the parser. Then structure is equal to parser dot get structure one fat, and the file name is one fat dot pdb. Okay. Um, then for model in structure, then for chain in model, we will print chain ID, chain dot ID, right? And then let's just look at the first like five residues of the first chain. So residues is equal to list chain dot get residues. Then for res in residues, for five elements, we will do this five uh, print residue res dot get there should be something like, yeah, res name. Then after that we will not close it, we will add uh, res dot id uh, 1. Yeah, okay. Notice how this how we drill down through the layers in this loop. So let's just try to run it and see what happens. And yeah, as you can see, uh, we have seen the first four of these. Some warning is there. We can ignore it for now. So yeah, uh, we start at the model, then move to the chain, and then list the residues. This allows us to target very specific part of a protein. So the real power comes when we reach the atom level. So every atom in a PDB file has a coordinate. So let's find the location of the first carbon alpha atom in our protein. Right. So for that, what we will do is we will select the first residue in the first chain. So first residue is equal to structure 0 uh, A 1. Then we will get the carbon alpha atom. Ca atom is equal to first residue, and then we will write Ca here. Right. Let's print this atom Ca atom dot get name function. Okay, and after that we will print the 3D coordinates. So 3D coordinates Ca atom dot get vector, and let's try and run this, and here you have it. This vectors gives us the exact position in angstroms. Because these are just numbers, we can use math to solve biological question. So next part is measuring distance, which is like like a common question in bioinformatics is how close are these two parts of the protein. So BioPython makes this incredibly easy. To find the distance between two atoms, you just simply subtract them. So let's get two different atoms first. Rest one is equal to structure 0 A 1 Ca. Right. We will copy this. We will name it rest 10 and then okay 10 here and Ca. Okay. Now we will calculate the distance in angstrom. So distance is equal to rest 1 minus rest 10. Simply that. And let's see what happens. Yeah, we will print also the distance between residue 1 and residue 10 is distance dot 2 for limiting the difference to two floating point values, and then angstrom. So here you have it. This simple subtraction tells us the Euclidean distance. This is the foundation for finding active sites, predicting where drugs might bind, or studying how a protein folds. So we have just scratched the surface of structural bioinformatics. We went from a text file to navigating 3D space and measuring molecular distances. Understanding structure is the key to drug design and understanding many diseases. So in our next videos, we are going to stay with proteins, but move to a larger scale. So all the code and link to the files will be in the description. And if you are excited about the 3D side of biology, then hit that like and subscribe button. And if you manage to calculate the distance between two atoms in your own protein, let me know in the comments. See you in the next video.